Hi everyone, this is the review video for the end of course exam. In this particular one we're going to cover DNA structure. Now this is not all we need to know about DNA, but this is the structure of the DNA and how the DNA, what the DNA's job is and part of how it functions. The first thing we're going to talk about is what exactly does DNA stand for. Well, it's a big long word, deoxyribonucleic acid, but you can see we take the D of deoxyribo, the N of nucleic, and the A in acid, and it puts us three letters DNA. Now here's something that we know. Okay, we know that DNA is an acid. Now I don't want you to think there's a jar of DNA somewhere that you can pour onto like your cat and it'll dissolve. That's not how it works. It's an acid simply because of its structure. It's an acid simply because it is has a certain group hanging off the side of it. It doesn't necessarily make it caustic or acidic. We also know that it has a ribose. That's what the ribo in ribonucleic acid means. It has a ribose sugar. And so when we look at the structure of it in a few minutes, you're going to see that it's made out of a sugar called ribose. But this is a very special sugar because it is missing an oxygen compared to other ribose sugars, so we call it a deoxyribose. So it is a nucleic acid that has a ribose sugar as a backbone, but that ribose is missing an oxygen compared to other ribose sugars, so we call it deoxyribose. So it's a nucleic acid made out of deoxyribose. We call it deoxyribonucleic acid, or DNA. Now, as with all things in science, the only reason that deoxyribonucleic acid or deoxyribose is used is because of its shape. It has the exact right shape for doing what it does, and it does something pretty amazing. If its shape were to change very slightly, for instance, if the oxygen were to be back in there, it would have a slightly different shape, which means it has a slightly different function, or it doesn't function at all. If instead of using the ribose sugar for the nucleic acid, it were using some other sugar, it couldn't work. Okay, so DNA, or deoxyribonucleic acid, uses a very specific structure to do its job. And if it changes at all, then it doesn't work anymore. So let's talk about what its structure or what its function is. Because of its structure, it has a function, and its function is to build a structure called DNA, a very long polymer chain of nucleic acids that goes together to build your DNA, or your DNA molecule, which is a very long molecule. And in that molecule is a specific code. Okay, now when you think of a code, I want you to think of, Morse code, or I don't know, any other kind of code. But if you think about it, reading a book, that's code. It's a series of letters that go together in a way that you can understand. That's all DNA is a series of nucleic acids that go together to create a very specific code. The code itself doesn't do anything. Think of it like instructions for building something. I think there's a video in a minute where we talk about an IKEA, I don't know, a table or a desk, right? Now, if you ever seen one of those or anything like this, you get a little instruction booklet. The instruction booklet isn't anything; it's just the instructions, actually. But it tells you how to take the flat pack materials and all the little screws and stuff, and to put it together to create the table. That's all that the DNA does, is it tells us how to make something called proteins. Now, proteins are very important. Proteins are everything to us. Our entire body is made out of proteins. Our entire body is shaped the way that it is shaped, and colored the way that it is colored, and etc., because of the proteins that are there. And in fact, the difference between me and you and your brother, you and the kid across the street, you and some kid over in Africa or India, the only difference between you guys is the types of proteins used to make you versus the types of proteins used to make them. For example, this person has blue eyes. That's because they have actually less of a protein than someone with brown eyes has, but it gives you a difference. It gives you a trait. So what I want you guys to first understand is that DNA is just a code to tell us how to make proteins, which then give us traits. And this process is rather complex, and it's going to take us several videos to talk about. So today we're just going to talk about the structure of DNA and one of its functions. Okay? Take a long, hard look at this picture. This picture explains to you a lot of things. Okay? First of all, it tells you what we're talking about in terms of the cell. Inside of the nucleus of the cell are chromosomes. Okay? If you were to take, humans have 23 pairs of these. If you were to take a look at this chromosome, really zoom in close, you would see that it's made up of a very long strand, and I'm going to show you a video of this in just a second. And if you were to just keep zooming in on that strand, you would find out that all that a chromosome is a bunch of DNA all coiled up around these little things called histones, but coiled up. That's all it is. DNA coils up to make chromosomes. So I want you to first make that connection. DNA coils up and coils up and coils up to make chromosomes. Chromosomes are found in the nucleus of the cell. Okay? The next thing I want you to understand about this picture is that this is a real, you can imagine how long this is. See these base pairs here, this GC pair or this AT pair, those are called base pairs. That's the code, and you have 3 billion of them to make you up. So every nucleus of every cell in your body, not every cell, some of them don't have a nucleus, but of all the cells that have a nucleus in your body, inside of it is at least one copy of this DNA, 3 billion letters long. If you were to try to read one of these, if you were to try to read this DNA 1 pair per second, 1 second, 2 second, 3 second, 4 second, at that pace it would take you 75 years non-stop of looking at it before you could see the entire structure. It's huge. But the thing is that's not the whole thing. The whole thing is not just the base pairs, because sections of this thing sliced up are what we call a gene. Now a gene is what makes a protein, and proteins give us our traits. So all along here are some blank spots. Some of the DNA is just blank, it's useless, and it's not really useless, but it's blank. There's spaces in between for a reason. But then every once in a while we hit a string of the DNA that makes a gene, and the genes are what makes the proteins, and that's what's important. Now this structure is amazing because it can self-replicate easily without error, and that's what we're going to be focused on today. We're going to take a look at this video that's going to explain this a little bit better right here, and it's not going to have any sound, so I'm just going to talk over it. But this is a piece of DNA. It's magnified 100 million times. It's unbelievably small. But right there, see those little spots right there? Those are the base pairs. Now, at a certain point in DNA, we want to pack it up. So normally it's all uncoiled, but now we want to start coiling up, because we're going to pack it up to move it. So it starts wrapping around these purple things here called histones, and it creates a structure called a nucleosome. Okay. But then it starts wrapping in on itself again. So now it's coiled once around the histones to make the nucleosome. Now it's coiled up again to create another structure. All right, and so it's just coiled and then coiled, and this is called a chromatin fiber. And now it's going to coil up even again, right? So it coils up for the third time now. Now we're at 10 million magnification. We're 10 times zoomed out than we were a second ago. And so now we're all coiled up, and guess what it's going to do? It's going to coil up yet again. So we have coiled and coiled and coiled and coiled and coiled. Okay, now we're looking at 1,000,000 times magnification, but this gives us the structure that we call a chromosome. And I guess it's going to go away. Hold on, there we go. Well, let's do it again. This creates a chromosome structure. Go away thing. And then that is found in the nucleus of the DNA along with all the 22 other chromosomes, and it starts a process called cell division, and we've only magnified 3,000 times here, so we've zoomed out, right? And then it goes through prophase, metaphase, anaphase, and telophase, and then we get 2 new things. But it self-replicates. Why does it self-replicate? There's one purpose for replication, and that's to make an exact copy. And here's the deal: every cell has to have an exact copy. So as a result, when I create a new cell, it's got to have its own copy. So before I can create a new cell, I have to copy all the DNA. And the structure of the DNA is designed so that it can be rapidly copied. 3 billion pairs. If you were to look at it 1 per second, it takes 75 years for you to see it. Can you imagine how long it takes how long it would take to copy it if you did it one at a time? It takes 75 years. We need it done in about, I don't know, 7 minutes and 50 seconds. And so this has to be done very quickly, and by the way, can't have any errors, because an error means that we're no longer making the right protein, and who knows what the heck's going to happen. So this is an incredible process that happens very rapidly without error, and it's because of the structure of the DNA. and I got to get past this. It's because of the structure of the DNA. Now, this was discovered in the 1950s by two guys, James Watson, right? Yeah, Watson's the younger guy. No, not Watson's the older guy. James Watson here and Francis Crick here. Now, they were working at a college in London, King's College, and there were a lot of people trying to figure out about heredity. Now, we knew all about heredity because 100 years earlier, guys like Gregor Mendel and Charles Darwin had said, that heredity is the key to understanding all of biology. But it took a long time and a lot of experiments by a lot of other people like Chargaff and people who did experiments that led up to what Watson and Crick finally put together. They put together they mathematically figured out the structure of the DNA molecule. Lots of people had been trying, lots of people had realized that it probably is DNA that holds the heredity or holds genes, but what does it look like? And these two guys won the Nobel Prize for it. A quick little funny story, they were working next door to a lady named Rosalind Franklin, who was also trying to find the same thing, and they were colleagues, but they were kind of racing each other because they knew that whoever figured this out would win the whole thing, win all these prizes and stuff like the Nobel Prize. And one day while Rosalind was at lunch, they snuck over into her office and took a quick look at something that she did called X-ray crystallography. And long story short, they realized that the DNA molecule was an even width the whole way down, and there was only one way that could happen, and that was with using these base pairs. So they put it all together, and they stole from Rosalind, and they published a one-page paper the next day, and that's how come we call it the Watson and Crick model of DNA, even though there were other people that were just literally days behind them. We don't even know their names because, they were second. The thing is that these two claimed, and I believe it, these two claimed that they weren't trying to do anything hateful to Rosalind because, back then in the 50s she was a woman scientist and that was just unheard of. But they claimed they weren't trying to be hateful to her, but she got very mad. And after they got the Nobel Prize, Rosalind actually died a few years later, and they went back and said we really should give her part of the recognition, and they did, and now she shares the Nobel Prize with these two guys for discovering the DNA molecule. Each DNA molecule is made up of millions of tiny subunits called nucleotides. Each nucleotide is, as I've already told you, it's a phosphate sugar, a phosphate, a pentose, and a nitrogenous base. And the pentose right here is the deoxyribose sugar. Okay, so these are the 4 nucleotides that make up DNA, and this is what's important. You don't really need to know the shapes or anything. You just need to know a few things. First of all, there are 4: guanine, cytosine, adenine, and thymine. Now I remember that by GCAT or GCAT. All right, guanine, cytosine, adenine, thymine. That reminds me of a few things. G and C always bond together. All right, they always stick together. And A and T always stick together. If I have a T on one side, there's an A on the other. If I have an A on one side, there's a T on the other. If something else happens, then we've got a mistake, right? So G's bonds with C and A's bonds with T. That's what you can remember from GCAT, okay? The other thing you need to remember are 2 terms called purines and pyrimidines. The purines have a double sugar. They have what's called a double ring structure. The pyrimidines have a single ring structure. Now you can remember this however you want. I remember it as being the opposite of size. Purine is a small word, but it's the big molecule. Pyrimidine is a big word, but it's a small molecule. That's how I remember it. It's worked well since I was in college in 1990 something, but you can remember it however you want. But there are 2 pyrimidines, thymine and cytosine, and there are 2 purines, guanine and adenine. I also remember this by saying PUGA, PUGA. Which 2 are the purines? PUGA, PUGA. Guanine and adenine are the purines. Now these are just little mnemonics that help you remember stuff, but you really should remember this. As I said, they're supposed to only bond with what they call their complementary pair, and almost every time that's what happens, but there is room for error here. As you can see, there's a double bond between thymine and adenine and a triple bond between cytosine and guanine, but every once in a while guanine can stick to a thymine or adenine can stick to a cytosine, and then we got problems. That's called a mutation, by the way. This is the very basic structure. Here's that sugar called deoxyribose. There's a phosphate that makes the backbone, and the nitrogenous base. This is the guanine, the cytosine, or the adenine, or the thymine right here. Okay, and so most of the time we just kind of ignore this because it's always the same, but this is what's different. This base is what's always different for every nucleotide. And I threw one right here. You can see it right here. This is the backbone, the phosphate backbone, the sugar, and then the base. And over here you got the base, the sugar, and the backbone. Okay, they're kind of mirror images, but they stick together. Okay, and it's that way the whole way down. And if you could take this structure and copy it the whole way down, and mirror image it and copy the structure here the whole way down. But you will see how they stick together. A always goes with T, C always goes with G the whole way down. Okay, and then they twist up to create the helix. All right, now you can ignore things like major groove and minor groove and all this other stuff, because that's for if you ever decide to study biochemistry. But at a basic level, this is the structure of the DNA molecule. It has a left side and a right side. It's technically a polymer. A polymer is a bunch of monomers stuck together. Every one of these letters is a monomer. C's, A's, T's, and G's. And if I stick a bunch of them together, I get a polymer. This is actually two polymers. This side here is a polymer, and this side here is a polymer. And then they're stuck together. So it's a double polymer structure. And then it twists up. And this is so amazing because if we needed to make a copy of this, it's very simple. We just split it right down the middle, and I know that if there's an A here, what should I put there as a T? So I can make a copy of it. So this is easy to get if you'll just follow along. If I know there's an A over here, A always bonds with what? A T. If there's a T here, I know it goes here. If there's an A here, there's a T here, TA, CG, AT, TA, GC, CG, the whole way down. Now I just made this up. I just typed this out and just randomly picked it a little while ago. But this could be a gene inside of your DNA. It's a little too short. Genes are typically hundreds or thousands of letters long, but you can get the idea. This could be a gene. And on the other side, the complementary side, is the mirror image. TATAG, TACG, the whole way down. Okay? Now what would happen later on, and we're going to learn this on the next video, is that we can read this, because this is the code. We can read this and send these instructions out into the cell where there's a thing called a ribosome that takes these instructions and turns it into a protein. It's a process called transcription and translation, which is another video. But we can also make a copy of this. And if I'm going to make a new cell, I have to make a copy of this. This is just one of 3 billion letters. I have to copy all 3 billion letters exactly, or else I have a mutation. And it's not that hard to do. But it has to happen so fast that when something comes along and reads it, it's going in at the speed of a jet engine. It moves that fast. It is unbelievably fast, but it's also unbelievably accurate. Okay? Now, as we've talked before, a section of these letters is called a gene, and that gene contains the information or the code for making a protein. Each unique gene has a unique sequence of bases. They're not all written like this. That just depends, but it's 4 letters in some combination that creates a unique sequence of bases. The gene that I have for my skin color and the gene that you have for your skin color may have the same if you and I have the same skin color, or they may be different. But all it is the sequence of DNA. It then creates a unique sequence of proteins that then gives us our unique combination. There's nobody else in the world like me. I have a unique combination of DNA that I inherited from both of my parents. I have a brother who I share both parents with. He is my full brother, and he is the closest related person to me on the planet genetically, but he's different than me in quite a few ways because he inherited different traits from mom and dad than I did, and this gives us what's called our phenotype. And all it is what genes did I inherit from our parents and what genes did he inherit from his parents. And it created a person uniquely me and uniquely him. And we are very similar, but we have lots of differences as well. Now, this video is going to show us how all that works. Well, it's just going to give us an idea. So DNA actually works it's a big code, but the code is just the instructions. It's just like the instruction manual from IKEA. It doesn't do any good, but it's found in the nucleus, and outside of the nucleus are the factories. So the information is in the nucleus, and outside of the nucleus is where we build it. Okay, so it's like a factory. The information comes from inside of the nucleus, the code. Then that code is read, right? It's just a little piece of the DNA. It's not anywhere near all the DNA. It's just a little piece of the DNA, but that's a gene. That gene is then, a copy of that gene is brought out through a process called messenger RNA. We're going to talk about that tomorrow. And that's a copy of the instructions. And then there's a factory outside that takes those instructions and turns it into protein. It's fairly complex, which is why it's a whole nother video, but it's kind of like IKEA. You know, if you go to IKEA and you buy a table, it's not put together. Now there's a computer somewhere that has all the instructions to make everything in IKEA, but I don't need all those instructions. I just need one little piece of the instruction, the gene, to make the one table. So I don't, and by the way, they don't give me their only copy, do they? No, they make a copy of it and send it to me. And then I take it home and I put together the table. The table is the goal. The table is like the protein, but the information to do that is what we're talking about today with DNA and RNA. And proteins don't last very long, typically a couple of days. So we've got to constantly be making proteins. You are literally making billions of proteins right now as we speak, and they do all kinds of things. They build our brains, they build our body, okay? They make our stomach or our muscles. They also tell us what to do. They transmit information from one cell to the other. They also help us to digest things. It's an unbelievable, our body functions basically around proteins. But this process that I'm talking about now is how we make those proteins, okay? How do I get to the next slide? So the code works like this. This is just a very small piece, but TACTAT may give us purple flowers, and TACTAC may give us white flowers. One letter difference in that gene will give us purple versus white flowers. Now remember, the gene is actually much longer than this. It keeps going that way. So think of bases of DNA like letters. Letters form words, words form sentences, and everything I've ever read was in English, and it was made out of the same 26 letters. But you know, go into a library and look at all the books. I mean, that's 26 different letters made everything in that library. Well, there's only 4 genes, but you can put, or 4 nucleotides, but you can put them together in very long combinations to create as many possible genes as you can think of. And think of every trait you've ever seen on any living thing, not just humans, and it's all made with the same 4 letters. I have fairly white skin because, you know, I'm a white guy living in Florida, which is a problem. I just had to go to the dermatologist, but I have very white skin. Okay. Think of a tree. Does it have white skin? No, it has bark and leaves. But for me to make skin, and for that tree to make bark, and for the armadillo to make a shell, and for the fish to make a scales, and for mushrooms to make whatever mushrooms have, you know, it all uses the same 4 letters, just in different combinations to make different codes. And that's how DNA works. Now, next video that's the end of it. So the next video is going to be about how to take the code and how we turn it into proteins. But we're going to stop here on this one and go back and review this as many times as you need to, and we're going to work on the next one, and I'll see you on the next video.